So Ramapithecus, it lived 3 to 4 million years ago, and Ramapithecus were more of a man-like, and they were probably not taller than 4 feet. The primary producers are larger in quantity, and then comes lesser in quantity the consumers, and it goes on decreasing. So in dichogamy there are two types. One is protoandry and protogyny. So protoandry, the male flowers will mature first and the females will not mature. Hello everyone, welcome to the second session of the model question paper discussion for second PUC Biology. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So looking into the recap, that is what we learned in the first question. So in the first question, we had learned the Part A and Part B. That is, we had solved the MCQs, the fill in the blanks, and we also solved the 2 marks question and answers. So moving further in this session, we will continue with solving questions carrying 3 marks and question carrying 5 marks. So let's begin the session. So this session has Part C and Part D, where in Part C, it is answer any 5 of the following questions, and each question carries 3 marks. So the first question under this Part C, draw a label diagram of the fertilized female gametophyte and mention the ploidy of any one of the products of double fertilization. So we have to draw the diagram of the fertilized female gametophyte. So that is nothing but diagram of the embryo sac. So we need to draw the diagram of the embryo sac, which contains 7 cells and 8 nucleus. So in this, because this is a question for 3 marks, at least 6 labeling should be there. So one of the labeling, that is the chalazal end and then the micropylar end. So you can draw it anyway. In the exam, you get confused between the chalazal end and the micropylar end. That is whether the chalazal end should come at the top, the micropylar end should be at the bottom, or should it be vice versa. You don't have to worry about that. Any way you draw it, always remember where there is the where you draw the, or if you mention the micropylar end towards the top, then you have to draw the synergids and the egg apparatus at the top. So you have to always remember antipodals are present always towards the chalazal end, and the synergids and the egg cell are present towards the micropylar end. So first marking chalazal end, then the antipodals, that is 3 antipodals that are there. Then comes the central cell, which is having 2 polar nuclei, having a ploidy, that is of haploid. And then the egg cell, then comes the synergids, and in the synergids, the finger-like projections called the filiform apparatus. So there needs to be 6 markings, and if the diagram is drawn very neatly and labeling is not done, then marks will not be given for the diagram. So at least 6 labeling should be there for a 3 marks question. So the continuation of this question, so we have finished drawing the diagram. So the next question is, mention the ploidy of any one of the products of double fertilization. So here, what exactly involves in the process of double fertilization? It is the egg cell, egg, and the central cell. They are the one which involve in the process of double fertilization. So egg, which is again haploid, for this one of the male gamete will fuse to form a zygote, which is diploid. So therefore, diploid zygote is one of the end product of double fertilization, and one more male gamete that is there, that will enter into the synergids, that will go and fuse with the polar nuclei. So in polar nuclei, there are 2 nucleus, therefore N plus N, and for this one more male gamete having a haploid nuclei will go and fuse, therefore resulting in the formation of a primary endosperm nucleus, which is nothing but triploid in nature. So therefore, what are the 2 products? One is a diploid zygote and the other one is a triploid primary endosperm nucleus. So moving further, the next question, parturition is induced by complex neuroendocrine mechanism. Comment. So they have given a statement here. That is, because of complex neuroendocrine mechanism, parturition happens. What is the exact reason for that? We need to comment on that. So parturition is a neuroendocrine mechanism, and it is started by signals which are generated by the fetus and the placenta. That is, once the fetus becomes fully matured, they start to induce parturition. And this actually results in fetal ejection reflex. This fetal ejection reflex causes release of oxytocin. So oxytocin, again, it will induce fetal ejection reflex. Again, more amount of oxytocin will be produced. Again, it will induce fetal ejection reflex, and therefore the birth of the child will happen. So that is what. So for contractions to happen, this oxytocin is, or for strong uterine contractions to happen, oxytocin is very, very important. So we have to mention about that here. Moving further, the popular and effective contraceptives include IUDs. So they are talking about IUDs here. So the question is based on IUDs, that is intrauterine devices. So mention the types of IUDs with an example. So you need to always read the question properly. Just mentioning the IUDs and leaving it just like that will not fetch you full marks, because for each of the IUD, they have specifically asked to mention one example each. So the first type of IUD is copper releasing IUD, and the example is multiload 375 and copper T. Next is hormone releasing IUD. Example is LNG 20. Next is non-medicated IUD, that is Lupus. Loop is the example. So 3 you have to write. So for each carries half a mark. So therefore a total of 3 marks. So if you don't write the examples and you just mention the IUDs, then you will get just 1.5 mark. If you write the examples, everything, you will get a total of 3 marks. Next question. Write the salient features of the following human ancestors. So they have given the following ancestors here, that is Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Neanderthal man and Homo erectus. So you have to write points for each of this. So Dryopithecus, they lived about 15 million years ago. They were hairy and they walked like gorillas and chimpanzees, and they were more ape-like. So Ramapithecus, it lived 3 to 4 million years ago, and Ramapithecus were more of a man-like, and they were probably not taller than 4 feet, but they walked upright. So therefore, they looked almost like a man, that is Ramapithecus. Next, Australopithecus, they lived around 200 million years ago, and they hunted with stone weapons. But though they hunted with stone weapons, and the major staple was fruits. Next is Homo habilis. Homo habilis, they are the first exact human-like beings, and they had a brain capacity of 650 to 800 cc, and they were not meat eaters. They did not eat meat. Then Neanderthal man, the word itself says Neanderthal. They used to move from places to places, so therefore they needed protection according to the environment. So therefore they protected their body with hide, and also they buried their dead. Next is Homo erectus. Homo erectus, they probably ate meat, and they lived near East and Central Asia between 1 lakh to 40,000 years ago. And they arose in Africa, and they moved across or spread themselves across different continents. So that is how different races of humans developed. So next question, describe any 3 properties of cancerous cells. So any 3 properties. So the first property is cancer cells, usually they don't have a proper cell division. The cells divide in a haphazard manner, that is uncontrolled division is there. So therefore uncontrolled growth of the cancer cell leading to tumor. Next is cancer cells have the property of invading the neighboring tissues, and that is capable of metastasis. Next is immortality, that is cancer cells are, the death of the cancer cells is very rare because they keep on dividing. It is very difficult to control them. They have immortality, that is, the death is not possible. So these are the 3 properties of cancerous cells. Next question. There is a table that is given below. We have to complete it with appropriate answers. So in column 1, they have given name of the microbe and they have given A, E, wherein we have to fill what is it. And in the second column, they have given name of the product. And in the third column, they have given the uses. So looking into the first column, so they've mentioned the name of the product. It is lactic acid. So lactic acid is produced by lactic acid bacteria. So what should we fill for A? Lactic acid bacteria. Or in the exam, you can just, instead of doing the column and wasting your time again, you can just write A, what it is. You can then go for B, what it is, C, like that you can write it down. So lactic acid bacteria produces lactic acid. What is the uses of lactic acid? The use of lactic acid is it increases vitamin B12 content in curd. Increases vitamin B12 content in curd. Or instead of writing lactic acid bacteria, we can also write it as lactobacillus. That's also same. So next one. Coming back to the second row, they have given here methanogens. They have mentioned the name of the microbe, methanogen. Name of the product, methanogens, it's biogas. Uses, they can be used for cooking purposes. Next is name of the microbe they have not given, but they have given here treatment of bacterial diseases. So what would be the name of the product? It would be antibiotic. Now, if I say antibiotic, if I write antibiotic here, can I name the microbe? Not possible, right? It's not possible. So, I have to specifically mention which antibiotic. So, antibiotic, penicillin. Now, I can write the name of the microbe here for E. It will be Penicillium notatum. This is how we have to fill it. So that's what A is for lactobacillus. B, lactobacillus increases vitamin B12 content in milk, and methanogen bacteria produces methane. They are used as a biogas, and penicillin is the product and it is obtained from penicillium is the microbe. So moving further, question number 34, they have given the question as represent diagrammatically the pyramid of number in a terrestrial ecosystem. So we have to draw the diagram as it is in the NCERT. So pyramid of number means it is an upright pyramid wherein we can't write here, mention joules and all that. Why? Because the diagram is not asked for pyramid of energy. It's asked for pyramid of number. So here primary producers 58,42,000. Next is the consumers. The primary consumers are 7,08,000. Then comes the secondary consumers, which is 3,54,000. Then comes the tertiary consumers, which are 3 in numbers. So therefore here the primary producers are larger in quantity and then comes lesser in quantity the consumers, and it goes on decreasing. So therefore it is an upright pyramid. So you have to draw this, and some students what they do is they'll just draw this and mention the numbers and leave it. No, you have to write the primary producer, the primary consumer, everything needs to be mentioned if you need to get a full mark. So this was about part C. So next we'll move on to part D, which carries 5 marks question. And again part D is split into 2 mains. So in that the first main of part D, that is answer any 4 of the following questions, which carries about 5 marks each. So the first question under this, flowering plants have developed many devices to discourage self-pollination. That is, we had learnt about the incompatibility, how to prevent pollination or to prevent self-pollination. There are some outbreeding devices, so we have to mention them. So that is to promote cross-pollination and to prevent self-pollination. So first one is self-incompatibility. So self-incompatibility, it is a genetic mechanism wherein either the pollen will not mature. So if the stigma matures and the pollen is not released, then there will be no chances of self-pollination happening. The other thing is sometimes the pollen grains would have been released, but the stigma would not be mature enough to receive the pollen grains. So that is called as self-incompatibility. So that is one point which you can write. The next one is about dichogamy. Dichogamy, the separation of the male and the female reproductive organs in time. For example, there are two types. That is one, for example, protoandry is what I have mentioned here, and also protogyny is also mentioned. So protoandry means when the word itself says proandry, that is the male parts will mature first, andrecium. So in dichogamy there are two types. One is protoandry and protogyny. So protoandry, the male flowers will mature first and the females will not mature. So therefore, when the female parts are not mature, there is no chance for self-pollination to happen. So protogyny is when the female parts mature first and the male parts are not mature enough. So next we can write about hercogamy. So hercogamy is nothing but the arrangement of the female and the male reproductive parts. So that one also we can mention. Next is heterostyly. So heterostyly is the length of the stamen. Say for example, the stigma is located very high and the anthers are located below, then the chance of pollen grains falling on the stigma from down going up, it is not possible. So that is about heterostyly. So that also you can write. Next is pollen release time. When the pollen is released, the stigma is not receptive for the to receive the pollen grains. Next is hypogynous flower. That is where the gynoecium is higher than the andrecium. So the gynoecium is placed higher above, then the chances of pollination are quite very less. So all these you can write. So 5, if we write, it is sufficient. So next question, draw a labeled diagrammatic sectional view of the human female reproductive system. So the female reproductive system, again this question carries 5 marks. So at least 10 labeling needs to be there. And do not draw the diagrams very small. At least let it cover half a page and let it be neat enough. And while marking also, what most of the students do is they don't mark it properly. Say for example, if this is infundibulum, they would have taken the arrow mark till here. No, you have to, when you put the labeling also, when you draw the lines also, you have to be very, very careful about it. So here we can write about the layers of the uterus, that is the endometrium, the myometrium, the perimetrium, then uterine cavity, uterine fundus, the isthmus, then comes the ampulla, then comes the funnel-like region which is called as the infundibulum. Then you can mention about the finger-like projection which receives the ova or the egg, that is the fimbriae. Then you can write the ovary, then the cervix, cervical canal, vagina. So totally 10 labelings need to be there. So moving further, few autosome-linked recessive gene blood disease occur in human population. Among them, some are related to qualitative and some are related to quantitative problem of synthesizing blood proteins. Explain. So, autosomal recessive disease are mainly caused by mutations that happen in the gene, either because of usually point mutations, and this mutation results or affects the synthesis of the blood protein. So, these mutations can cause qualitative or quantitative problems which affect the structure and function of the hemoglobin. So, one of the best example that we can take here is of sickle cell anemia. So, because of one mutation happening, there is an entire change in the amino acid chain. So, therefore, it results in sickle cell anemia. That is where the RBC should be oval in shape almost, it starts to look like a sickle. So, qualitative problems alter the structure or function of the blood protein, making it dysfunctional, whereas quantitative problem affect the amount of blood protein that is produced, leading to insufficient levels. So, what is qualitative here? Qualitative means the quality or the way the blood protein looks or the way the RBC looks will change. So, therefore, sickle cell anemia is one of the best examples. Quantitative means the amount of RBCs that is being produced. So, therefore, the quantity of hemoglobin will also come down. So, that is quantitative problem. For example, for qualitative problem, the best example is sickle cell anemia. Sickle cell anemia is a qualitative disease that causes improper synthesis of one of the globin molecules. So, this alters the structure of the red blood cell. which transports oxygen throughout the body. And thalassemia is an example for quantitative disease that causes less synthesis of globin protein, leading to the synthesis of abnormal red blood cells. That is, the number of red blood cells that will be produced will get drastically reduced. So that is about qualitative and quantitative. Easy to remember. Qualitative example is sickle cell anemia. It will convert the RBC into a sickle shape, change the shape of the RBC. Quantitative is thalassemia, wherein the number of red blood cells being produced itself will drop down. Therefore, the hemoglobin protein itself will reduce in number. So, moving further, DNA replication is fast, accurate, energetically expensive, substrate and enzyme dependent process initiated from specific site and cannot uncoil on its entire length. So they only have given, they themselves in the question, they have mentioned the properties of the DNA molecule. So what have they said? They have said that DNA replication is fast. They have said it is accurate. There no mutations will happen during the process of replication. And energetically, it is quite expensive because a lot of energy is required there. And substrates and enzymes are also involved in this particular process, and replication always begins at a specific site and it terminates as a specific site and cannot uncoil on its entire length. That is, the entire length of the DNA will not uncoil. Only it starts in the promoter region and the uncoiling ends in the terminator region. So that is what they have mentioned, the properties. Now they're asking us to explain the process of DNA replication, keeping all this in mind. So you have to write the answer wherein all these characteristics you have to mention here. So we'll look into the answer. DNA replication is a complex process that occurs rapidly with high fidelity or high accuracy at specific initiation points on the DNA molecule. Utilizing various enzymes are involved, various substrates are involved to create new identical DNA strand. That is, for the parental strand to create new daughter strand, all these are involved. And all this happens only when the DNA double helix unwind at localized or specific position, creating replication fork due to the high energy cost of fully uncoiling the entire DNA molecules. This process involves key steps like initiation at origins of replication, strand separation by helicases enzyme. Then for the daughter strands to form, we need a primer. For that, primase enzyme is involved. Then for the separation or for the addition of the nucleotides to the daughter strand or nitrogenous bases to the daughter strand, DNA polymerase enzyme is involved, and ligation by DNA ligase will happen to join the fragments of the lagging strand. That is, we know that there is one strand where the daughter strand that is formed is continuous, and on the other strand, the daughter strand that is formed is discontinuous. So the discontinuous strand or the Okazaki strands or the lagging strand needs to be joined. So for that, the DNA ligase enzyme is also involved, ensuring accurate duplication of genetic information. So all this has come in this answer. We have mentioned about the accuracy, where the replication begins, where it terminates, and what are the enzymes that are involved. Everything we have mentioned. So whatever points they have given in the question, all that is mentioned in this answer. So this is how you need to write. You can't just write simply. So what points they have given, what characteristics they have given of the DNA during the process of replication, everything needs to be mentioned in the answer. Moving further, next question. Some drug bottles had their name labels missing. So in a hospital, there were a few drug bottles. In that, some of the names were missing. So the staff needs to identify the drugs with their actions still written on them. But what is the function of the drug? What is it used for? All that is written. Only the name is missing. So we have to help. Let's help the hospital staff by giving names based on what the drug is used for, what it is effective against. Based on that, we shall provide the names. So they have given two columns here. One, they have mentioned the drug 1, drug 2, drug 3 and drug 4. And drug 5, here they have mentioned the effect. Why only these two are mentioned? Here, why didn't they give the name? Because the name label is not there in the bottle, but the uses of the drugs are there still in the bottle. So let's look into, by looking into the effect of the drugs, let's see what the name of the drug would be. So used by doctors as sedative and painkiller. So it's given here. We'll look at the answer here. So drug 1, it was mentioned in the bottle that used as a sedative and painkiller. So it is morphine. So the source is opium poppy or Papaver somniferum. So it's morphine. So morphine is used as a sedative and painkiller. So therefore the bottle should be named as morphine. So the next, drug 2, helps with insomnia and depression. So it is fluoxetine. So therefore the source is synthetic. It's a synthetic drug. Next is increases blood pressure and heart rate. So it is epinephrine. So the source is adrenal glands is the source. Next is acts as a depressant. So it is alcohol. The source is fermentation of sugars. Next is affects the cardiovascular system. That is etinolol. Source is synthetic. So like this we need to mention the name of the drug. So moving further, transgenic animals provide innumerable benefits to human beings. So what are transgenic animals? Transgenic animals are the animals whose genes have been modified for the betterment of the human. So we can use it for various purposes. So we need to justify the statement with common reasons. That is why transgenic animals are beneficial. So one thing is enhanced nutrition for improved human health. So transgenic animals contain nutrients or their products. For example, the milk might contain extra nutrients which is beneficial for the human health. Next is they contribute to environmental sustainability. Next is they are quite resistant to diseases when compared to the normal or those animals which are not genetically modified. So they have increased disease resistance. Next is they have higher productivity and efficiency in food production. So maybe say for example there is a transgenic chicken, so it might lay more number of eggs when compared to a normal chicken. So it's to give higher productivity. Or the cow might give more amount of milk when compared to the non-transgenic animal. So it's like that. Next is valuable models for biochemical research. So any medicine, before it needs to be tested on the humans, these transgenic animals can be used for testing the efficacy of the medicine for its side effects and all that, and then it can be distributed to the market. So next question. Explain the fascinating forms of interaction. So they have given two types of interaction. We have to explain about them. One is brood parasitism and the other one is sexual disease. Brood parasitism. So brood parasitism, it is the quail bird going and laying its egg in the crow's nest. So it is a type of parasitism where one species of bird lays eggs in the nest of another species, and the host bird, that is a crow, will think that it is its own eggs and she will take care of the eggs. That is, the host incubates and cares for the eggs of the parasitic bird. Here the parasitic bird is the cuckoo. So for example, the cuckoo bird lays its egg in the nest of the crow. So here the parasite is the cuckoo bird and the bird subjected to brood parasitism is crow. That is, crow is the host. So the thing is that because this has been running around ages, the cuckoo bird's eggs now have started to look exactly similar like the crow. So the crow cannot distinguish which is her egg and which is the cuckoo's egg. So this is about brood parasitism. Here the parasitic bird is benefited. Why? Because using enough amount of energy to build a nest, it can directly go and lay its eggs on the crow's nest or which has been built already. So therefore, it is benefited. Next is sexual deceit. So sexual deceit is a type of mutualism where a flower mimics a female insect in order to attract a male insect for pollination. So the flower petal will look exactly like a female bee. So in color, in scale and in markings as well. So the best example is the bee orchid, that is Ophrys apifera. It uses sexual deceit in order to attract the male bees so that it can get itself pollinated. So sexual deceit is used by the bee orchid in order to get itself pollinated. So next question, that is the next main under part D. So here they have given, so these looks like competitive questions. So a puzzle kind. So we need to be very careful in solving it. So under this section especially. So results of Mendelian dihybrid cross are represented in the form of Punnett square. So I have given the answer here. We'll look into the Punnett square first. So this is the Punnett square. So you all have learned about the dihybrid cross, the monohybrid cross and all that. Now you don't have to do it because it's already given in the question paper. So we have to answer accordingly. So what is the first question we shall look into? So what is the first question we shall look into? So we have to find the number of parental types progeny. So let's begin. So number of parental types. Which are the parental types here? These two. Capital Y, capital Y, capital R, small y, small y, small r, small r. So what's the first question? We have to find the offsprings that exactly look like the parent. So therefore the offspring should have yellow round seeds, right? And it also should have green wrinkle seed. So let's look into. So here there is one and there is another one, because these two are the only ones which looks like the parents, exactly like the parents. So therefore the answer is 2. 2 is the answer. So therefore for this, what is the answer? 2. So moving to the next question. Number of recombinant progeny. So recombinant progeny, that is it. So what should it have? Because it is a recombinant, both these should recombine. So it will have capital Y, capital R and small r. So now let's look into the progeny which is having this. So capital Y, small I, Y, capital R, small r. Here, 1. Next, there is one more here. So there is one here, 2. Then here 1, 3. And here. So these are recombinant. So the answer will be 4 for this. That's 4. Next one. Number of homozygous recessive. So homozygous recessive means it should have small y, small r and small r. That is homozygous recessive. So let's see how many are there. So homozygous recessive, small y, small r, small r. So only one is there. Only one. So therefore, the answer is 1. Next, number of homozygous dominant progeny. Homozygous dominant progeny means it should have capital Y, capital R. That again is just 1. Only one is there. So therefore, the answer is also 1 for this. Next is number of homozygous progeny for both the traits. Number of homozygous progeny for both the traits. So they have not mentioned homozygous recessive progeny, homozygous dominant progeny, nothing like that. They've just mentioned homozygous progeny for both the traits. So it should have a YYRR and a YYRR, because both traits should be homozygous here. So therefore, how many here? It is again 1 and 2, just 2. So therefore, the answer for E is 2. So next, number of heterozygous progeny for both the traits. Heterozygous means it should be YYRR. So heterozygous for both, that is for yellow and round, it is heterozygous. So we'll count how many are there. That is YYRR. Again, it's 1, 2, 3 and 4. So the answer for this is 4. Next question, number of pure line progeny. Pure line means it should look exactly like the parent. So therefore, it should have YYRR and YYRR, because these were the alleles of the parents, right? So that's why. So therefore, we had counted earlier itself, it is just 2 in number. So therefore, the answer is 2. So next question, number of homozygous progeny for single trait. They have not mentioned here dominant or recessive. So we can take both the color and the shape also. But here, one of the traits should be homozygous. So first we can take YY and RR. then we can take YY and R. So this can be considered. So we have to take both of these. That is, we need to search for capital Y, capital R, small r, and capital Y, small y, and capital R and capital R. So how many are there we will look into? So capital Y, 1, 2, next capital Y, small y, and RR. This is the third one here, fourth one. So therefore totally 4 are there. So the answer is 4 for this. It's 4. Next one, number of heterozygous progeny for single trait. Heterozygous progeny for a single trait. So let's see heterozygous progeny. So heterozygous progeny means I have to consider capital Y, small y, and capital R, and one more capital Y, capital R, small r. So that's also the same count. If you count, we'll get the same thing because it is one and the same here. So it is 4. Next is number of recessive progeny for single trait. So recessive progeny for a single trait. So recessive progeny. So the trait that I will consider here is yellow. So YY can be R, or it can be YY and R. So how many are there? We will look into. So small y, YY, RR, it should be YY, R and R. So therefore, it is 1, 2 and 3. So the answer is 3 for this one. That is J, it is 3 numbers. So moving further, given below are sequences of nucleotides in a particular mRNA and amino acids coded by it. That is AUG stands for methionine, UUU stands for phenylalanine, UUC for phenylalanine again, GAG for glutamine, UUA for leucine, GUG for valine, UAA, and so on. So write the properties of genetic code that can be correlated from the above given data. We have to write the property of the genetic code. So here the codon is triplet. So sometimes they give you this just to make you confused. So the entire question is about you need to write the properties of the genetic code. So they have given 4 or 5 properties in the textbook, NCERT textbook, right? The same thing you need to write down. So the codon is triplet. That is, it is always mentioned in 3, right? So therefore it is a triplet, and total 61 codons code for amino acids and 3 codons do not code for any amino acid because they function as the termination codon or the stop codon. Then some amino acids are coded by more than 1 codon. Say for example here itself, phenylalanine is coded by UUU and also it is coded by UUC. Next point, the codon is read in mRNA in a continuous fashion. There is no comma or full stop in between. It is read in a continuous fashion without any punctuations. And the next point, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine amino acid itself. That is, whatever it is, that codon codes for a specific amino acid in all the organisms in this planet. And AUG has dual functions. That is, it codes for the amino acid methionine and also it acts as the start codon or the initiator codon. Next is, there are 3 terminator codons or stop codons, that is UAA, UAG and UGA. So these are the properties of the genetic code. By giving that, you might get tense, like what is this? Should we do something with what is given in that genetic code? No, it's nothing like that. They have given it just to confuse. It's just that you need to write the properties of the genetic code. So moving further, given below are the diagrams of plasmids A and plasmid B. Observe and answer the question that follows. So what are the questions we will look into? So which plasmid is or are you select for cloning and why? Which plasmid would you select for cloning? What is insertional inactivation? Will the number of culture plating you should make is same or different to select recombinant if insertional inactivation is possible? Comment. So we'll look into the answer. So you have to look into these two and say which one is the better plasmid. So the question 1, which plasmid would you select for cloning and why? So for cloning, plasmid B is preferred because it contains lac Z gene which allows for blue or white screening. So this helps in easily identifying recombinant clones. And the next question is, what do you mean by insertional inactivation? Insertional inactivation refers to the process where the insertion of a foreign DNA fragment into a plasmid disrupts a gene, rendering the gene inactive or making the gene inactive. Next question was, will the number of culture plating you should make is same for both the plasmids or different? So if insertional inactivation is possible, the number of culture plating should be different, as you would need to plate both the transformed cells that contain the plasmid with the insert and those without to identify the successful recombinant. So this is the answer for this particular question. So with this, the session ends. That is the session 2 for the model question paper. So I hope you understood the session well and might help you for answering, like how you need to answer the questions and how much you need to be careful while reading the questions in the question paper. So this was about this session. Thank you.